Cell cycle. Again, this is the second video. We'll be discussing DNA and the behavior of the chromosomes. Cell division results in a genetically identical daughter cells, and cells divide millions of times to produce a mature organism. And this division, we all start from one single cell called as a zygote. The zygote divides millions and millions of times to form a mature organism. And each time it divides, it produces identical daughter cells. And they found that there are very few errors made during these cell divisions. And that means the cell division is very accurate. So to have this cell divisions with very few errors, the DNA has to be folded in a specific pattern, and the process has to be well organized. So before we go into the behavior of the DNA, let's look at the DNA molecule and how it is arranged in different organisms. In a prokaryotic cell, the DNA molecule, which is a double-stranded molecule, occurs as a loop, a circular loop, and it is in the cytoplasm called as the region where the DNA is found is called as a nucleoid. In eukaryotic cell, the DNA is actually located in the nucleus, and we have more than one DNA molecule, and the number each DNA molecule is called as the chromosome. Each DNA molecule is the chromosome. So the number of chromosomes vary from organisms to organisms. In humans, we have 23 pairs of chromosomes. That means we have 2 copies of each chromosome, one coming from each parent. In humans, most of the chromosomes, that is 22 pairs, are called as autosomes. These are the chromosomes responsible for most of the biochemical reactions in our body, which gives us the features and functions, whereas one set, the 23rd set, is called as the sex chromosome, which determines the sex of the organism. As I said, the chromosome the DNA molecule is very long, and it has to be folded in a specific pattern. So let's look at that. This is the organization of the eukaryotic chromosome. So the basic structure is that the DNA is a double helix. So 2 strands of the polynucleotides are bound together to form a molecule of DNA. Now this DNA molecule is wrapped around histone proteins. And those are the histone proteins. You can see green, blue, yellow, red. These are the histone proteins around which the DNA wraps itself. And each of this unit is called as the nucleosome. The nucleosomes further coils to form chromatin fiber. The fiber further condenses with a protein in the middle and condenses to form a specific structure. Again, I'm going to put an X on this. That means this is the form you will see in cells that are active or non-dividing cells. So in a non-dividing cells, the chromosome is in the form of chromatin. So the DNA is free for replication and transcription. Now, only when the cell is about to divide, the cells condense to form the chromosome. This is a condensed form of chromosomes. You don't see this until the cell is about to divide. Again, this is a picture of a cell which is ready to undergo cell division. How do we know? You can see this DNA is condensing. So let's look at what happens. How does the DNA organize itself when it's about to undergo cell division? Now what I did is, this one shows only one chromosome, but since we are diploid, I showed the pair of chromosomes, and this is 1A, 1B. So it's the same chromosome but two copies of it, and here it shows only one copy. Now if a DNA at this point condenses, it will have a center called as a centromere and the arms. Now you don't see this, but this is again a depiction what happens when you condense a normal chromosome. So a chromosome will have a centromere where the DNA is highly packed, and it will have the arm. Okay, so if I have to use this one, there should be another DNA molecule. So this is one copy, and so these are two of those, right? So now when the cell is about to divide, during the S phase, again this is not part of the mitosis, this is before the cell undergoes mitosis, the DNA actually is copied. So you have two. So you have one here, it becomes two. So that is one, those are the two. But these two copies are not separated. They are very close to each other. They're much more tighter at the right in the middle, called as a centromere. And that you'll see here too. That's a centromere. When the two copies are still attached, each copy is called as a chromatid. And you have these two are called as the sister chromatids because they are copies of the same chromosome. But this whole thing is still called as the chromosomes. Now during the cell division, these two separate. Once they separate, we call them as chromosomes. So as long as they're attached, each one is called as chromatids. When they separate, it's called as the chromosomes. So here I'm showing you that the chromatids separated out to form chromosomes. Okay. So once they separate, the DNA again uncoils and forms chromatin. Now this is how we started. And this is the final stage after cell division. So you have two, you made a copies and separated. This is this electron, scanning electron microscopy membrane, a picture. We can really clearly see this whole thing is called as a chromosome. And each one is called as a chromatid. And these two are the sister chromatids in the center, where the DNA is tightly coiled and more closely condensed, is called as the centromere. So we will talk about the cell cycle in the next video.